Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amirault. I'm the Senior Marketing Manager at Medicinal Genomics and proud member of the team that puts on the CanMed Summit. Well, our CanMed Advisory Board is hard at work reviewing all the great abstract submissions that we've received for CanMed 25. Our goal is to have all of our oral presenters selected by the end of the year and to announce them in January. So stay tuned for those announcements. We are also working on a number of great workshops that we will be announcing very soon. So if you have not already, sign up for email alerts at canmedevents.com so you don't miss any of those and other announcements. Also, if you are planning to join us at CanMed 25 next June in Puerto Rico, be sure to purchase your ticket package today and take advantage of our discounted early bird rate. Find more details at canmedevents.com. Our guest today is Dr. Nicole Gauthier. Nicole is a professor and extension specialist at the University of Kentucky. Her program focuses on disease management of specialty crops, including fruits, vegetables, and hemp. In her extension role, she develops educational and outreach programs to help growers manage disease through identification, understanding of pathosystems, and integration of management strategies. Her research program focuses on a range of hemp diseases, including Fusarium head blight, as well as stand establishment and seedborne transmission. Nicole's resources can be found at the University of Kentucky website, which I'll include a link in the show description so that it's easy to find. And she's also on Facebook and Twitter at KY Plant Disease, and on YouTube at Nicole Gauthier. Nicole's team at the University of Kentucky recently published a fact sheet about Fusarium in hemp plants, which caught my eye considering many of the cultivators we work with at Medicinal Genomics struggle with that pathogen. During our conversation, we discuss the financial impact of Fusarium on hemp and cannabis crops, the public health concerns due to Fusarium's production of harmful mycotoxins, how growers often misidentify Fusarium head blight as botrytis due to similar symptoms, leading to ineffective management strategies, environmental conditions that increase the likelihood of a Fusarium infection, challenges for prevention and management, including a lack of effective fungicides and resistant cultivars, and future research to better understand Fusarium spread, genetic resistance, and management techniques. Before we get to my conversation with Nicole, I'd like to thank this episode's sponsor, Rare Earth Genomics. Rare Earth Genomics is a dynamic partnership formed with Texas A&M AgriLife, one of the world's leading research institutions, to accelerate hemp research and create tools for farmers. Their mission is to increase the likelihood of a successful harvest for farmers through genetic analysis and rigorous environmental testing of vigorous cultivars. Learn more at rareearthgenomics.com. Okay, and without any further ado, please enjoy my conversation with Nicole Gauthier. Good afternoon, Nicole. Thanks so much for joining me on the podcast.
Hello, how are you?
I am great, and I'm excited to talk with you today about the work that you and your team are doing to better understand plant pathogens affecting hemp plants. Your team recently published a fact sheet about Fusarium in hemp plants, which we are going to dive into, but I know that's only one of many pathogens that your team studies. So maybe to start, can you provide a brief overview of the most common diseases affecting hemp crops, and why did you decide to focus on Fusarium this time?
Okay, so hemp in Kentucky and most everywhere is grown outdoors, primarily in the field. So when I look at cannabis in general, when I look at hemp or high THC, low THC, whether it's floral or grain or fiber, we essentially have the same plant, right? So I look at it from an outdoor standpoint. What pathogens can infect? Then we can go on the flip side and we can look at indoor cannabis or indoor hemp, however you want to phrase it. And an indoor environment is going to have its own set of pathogens because you have a very different climate system. So in plant pathology, most of our diseases are driven by not just the presence of the pathogen, but also the climate, right? Temperature and humidity. So in outdoor hemp, we see stand establishment problems. So we can have Pythium root rot, whatever the nature of the previous crop or the history of that field. So we could see a lot of seedborne seedling diseases like Fusarium crown and root rot, which is not Fusarium wilt. So the Fusarium solani based Fusaria. We can see a Rhizoctonia crown and root rot, and we can also see some Pythium root rots that can affect seeds or seedlings. When we get into a larger plant, everybody knows the leaf diseases. So we have a history of kind of all those leaf diseases. That's what interests everyone. And leaf diseases, Septoria leaf spot, Anthracnose or Cosper leaf spot, and then the big one, Bipolaris leaf spot. So those, we've been working on those for many years. I've been working on hemp since 2014. And then when we started seeing this head blight in grain hemp, there was so much talk about botrytis, botrytis gray mold, and that was like always the talk. And so everyone was seeing all of this fuzzy growth inside of their either their flower heads or their seed heads, and they were just automatically saying, "I've got botrytis, I've got botrytis," and knowing that pathogen, because it is ubiquitous, it affects many different kinds of plants. I knew it's too hot, it's too dry. And so we dove a little bit deeper, and that's when we realized that we had a very widespread disease. And not just the blighting, and so blighting is when it kind of turns brown, not just the blighting and the disease, but also the potential for mycotoxins from those pathogens. So that kind of took us down a road that we've done a lot of work. We're just starting to publish that work, but so yeah, we've been working with Fusarium head blight since about 2017.
Excellent. So was I hearing that correctly, that growers were misdiagnosing Fusarium as botrytis?
The danger of the internet. That's right. The danger of diagnosing by symptoms and pictures. So yeah, it was just kind of this blanket understanding that if it's fuzzy and white, it must be botrytis. And it was not. In fact, I rarely find botrytis in the field. Now I'm in a more southern climate than, so we could definitely see a lot more botrytis in the northern states, but we overall see botrytis more in the greenhouse. But any of that fuzzy growth I'm seeing in Kentucky, Midwestern states, mid-Atlantic states, and definitely south of me, it's almost exclusively Fusarium.
Interesting. And how does that affect how one kind of tries to manage the disease, if it's one or the other?
Well, both of the pathogens are true fungi, so they are fungal pathogens, and overall they need about the same temperatures and humidity to complete their life cycle. Botrytis likes very high humidity, so about 85% relative humidity. So it's got to be very wet. Botrytis is more of an opportunistic pathogen. So when it infects, it's infecting, it begins on something like necrotic tissue, maybe a dying leaf or something like that. And so it's kind of more opportunistic, whereas Fusarium is more of is pathogenic. It's highly pathogenic. So it can invade a healthy plant. Versus botrytis is more of that kind of humidity lover, and it needs you to open the door for it, so it can't just bulldoze its way into those plant cells.
Interesting. All right, well, that's a good segue. Can you describe how Fusarium affects hemp plants? How does it infect, and what is sort of the progression?
Okay, well, let's start with Fusarium is a genus, and it's a very large genus with a lot of complexes. So lots of species within a Fusarium complex. So most people, when they hear Fusarium, they think of Fusarium wilt, and that's Fusarium oxysporum, different different special forms, but Fusarium oxysporum or Fusarium wilt is a vascular disease. It starts in the roots, it's soil-borne, it moves its way up the vascular system, and it causes wilt because of just blocking water and nutrient uptake to the top of the plant and necrosis of the xylem.
Hmm.
Fusarium head blight is Fusarium graminearum. So that is infecting flowers and grain from the outside. It's a very, very different pathogen. And remember earlier, I talked about Fusarium solani, a root and crown rot. So there are three major Fusarium species that affect hemp or cannabis. But back to Fusarium head blight. The Fusarium head blight pathogens, we have identified 5. Fusarium graminearum is the common one. That's the one that you read so much that affects wheat and cereal crops. So Fusarium graminearum will infect flowers and grain. We have found it to infect growing tips and leaves, but primarily it's going to be a flower and grain disease.
Interesting. So different species of Fusarium will kind of attack the plant from different angles.
That's right. That's right. And to not just say Fusarium, because when we started working on Fusarium head blight, the assumption was that it was the Fusarium oxysporum, the Fusarium wilt pathogen. And they're very, very different, even though that first name, though that genus is the same.
Interesting. So what are some of the economic impacts of Fusarium on hemp cultivation?
So Fusarium head blight, as we call it, will blight out. And blight is kind of a rapid necrosis. That's what that kind of term means. Fusarium head blight will blight out a flower head or a seed head. So early shattering, more brittle flowers or grain heads, but we can have it at very low levels as well. So that's, remember I mentioned mycotoxins earlier. So Fusarium graminearum produces 2 different mycotoxins. So a mycotoxin is a toxin produced by a fungus. So mycotoxins, there's a history of mycotoxins in Fusarium species, the trichothecenes, for example, that can cause vomiting, digestion issues both in humans and in animals. So in hemp, in any feed, it's highly regulated. So even low levels of disease can cause high levels of toxins. And that's something that we're not regulating here in the US, not in hemp, not in cannabis. So everyone is really interested in looking at aflatoxin or some of those other toxins, which we're not finding, but we are finding those Fusarium mycotoxins from the head blight pathogens. So again, all the Fusaria out there, but that's our focus because that's what we're seeing the most, and that's what we feel is the most important, especially as hemp as an animal feed is coming onto market.
Yeah, so you mentioned that Fusarium could be there in low levels. So might it sort of escape some visual detection and have some of those mycotoxins present?
I'm going to spill the beans a little bit. My graduate student just came in with some year 1 data from 2 different fields here in central Kentucky, and he had Fusarium head blight at about 5% severity. So think about that whole seed head, and about visually 5% of that head had symptoms. So very low, something that a grower would probably overlook, something that I might have said, oh, well, and he ended up detecting DON, deoxynivalenol. He ended up detecting DON at levels that are above the threshold for human consumption in other grains. So of course we don't have that threshold for hemp, but that threshold that would have failed QC in a cereal grain market. And so we're finding those high levels of toxins, and in a year of drought and extreme heat this year, we had low levels of disease.
Wow. And so that is not being tested now? Those mycotoxins aren't being tested for in hemp products?
That is correct. That is
Wow.
correct. And so that was one of our delays in releasing some of our data. We wanted to double check and triple check and quadruple check because we didn't want to send a scare in the market. We wanted to make really sure that what we were seeing was a trend and it wasn't just isolated incidents. That is correct. We are not. We're looking at a lot of microbial testing
Mhm.
in hemp in some states. It seems like there's a huge interest in aflatoxin, right, and some of the other toxins, and we just don't have those regulations in hemp anyway, unless it's at a state level. But there are only a few states that are regulating those things, but they're not talking about fusarium toxins, especially not the trichothecenes.
Wow. Yeah, that was going to be my question. Where are those regulations coming from? Are they at a federal level or a state level?
Well, we could do a whole podcast on hemp regulations, right?
Yeah, and we
But
have.
no, some states are testing, and so
Mhm.
we hope that we can get the data out that's both going to help the hemp industry, not hurt the hemp industry, but also make sure that we are producing safe products for our end use, our end consumers, which could be animals or humans.
Excellent. All right. I think we've kind of set the stage for the stakes.
Yes.
So what about prevention? What are the best practices for preventing fusarium in different growing environments?
Well, that's the $1 million question, right? So what we know about Fusarium graminearum is the most common species that's causing Fusarium head blight. So if I say Fusarium graminearum, that's kind of the big one. So we know what we know about Fusarium graminearum and other head blight producing Fusaria we know from wheat and barley and cereals and corn. So we base a lot of things off of that until we can generate our own data.
Hmm.
So in wheat, so I'm going to give you a life cycle because, nerdy pathologists. So in wheat is a cool season crop. Fusarium comes in, it really kind of infects during anthesis, which is when that anther is kind of poking out of that wheat flour. And that's when infection occurs. And in Kentucky, for example, that's happening in May. We're harvesting wheat in June. And then we've got a hemp crop that really is not going in until July in most cases. So by the time hemp starts flowering and is ultra susceptible to fusarium infection, it's July, August. So how are we having this 2 month or more division between that wheat infection and that hemp infection? But we are. And what our research has told us is that in fields where there was wheat or near wheat, we have a lot higher incidence of disease and toxins. So there's a lot more Fusarium graminearum when there was wheat in the vicinity.
Hmm.
Um, and what we're finding out now is that wheat cover crops, even though they were terminated before they flowered, are still inducing a much higher amount of disease. So I don't remember your question now. So what
Prevention.
do we do about it? And that's what we're trying to figure out. What are all the roles? Where is it coming from? What we do know about fusarium, again, from the cereal grains research, is that those spores, those canidia, can get really high up into the atmosphere and they can move very long distances. So no one's safe, right? No greenhouse is safe if you've got a vent. But the closer we are, that proximity to any wheat or any other host plant seems to be really inducing a higher level of disease.
Now, is there any sort of sprays or bioagents that can be applied to kind of prevent an infection?
So right now, so I'm gonna back up just a little bit. Right now, we don't have any conventional fungicides or pesticides labeled for hemp. And we definitely don't for high THC cannabis. So we don't have any products anyway.
Mm.
In terms of organics, we've been looking at a lot of biologicals, and right now we're not finding anything that is effective. We have a few years of field studies, and we're trying lots of things. We're throwing a lot of things to see what sticks, and we just don't think we're getting anything right now.
Interesting. Okay. So one of my questions here, how does it spread between plants? But it sounds like it's airborne.
Airborne, yeah. So these are spores that can move long distances, and I mean, think about when disease spreads anyway, right? When those spores spread, it's usually wind-driven rain.
Hmm.
So if you kind of think about it, and we can look at, so kind of overlaying weather data with some of our spore trapping data, we can see that when there's a rain event, that's when we'll see spikes in disease. And that's really making a difference. And a drought year, a calm year, we can have a lot lower disease. But when we have a lot more rain, we can have a lot more disease. Because remember, fungi need at least 70% relative humidity, they need leaf wetness, and they need moderate temperatures. So things cool down when it gets cloudy. So even though it's hot and dry in the middle of the summer, if we got a couple of cloudy, rainy days, or even drizzly, it's enough.
Mhm. Yeah, that goes back to what you were talking about earlier of indoor versus outdoor. So these in an indoor environment, these are sort of conditions that you can control for, or you're not susceptible to the weather.
Yes, that's right. But greenhouses are a lot, or indoor environments rather, I'll just kind of lump those together. Indoor environments are a lot more humid, right?
Mhm.
Because we're not exchanging that air. The more we exchange air, the more we let things in. We have identified Fusarium head blight in greenhouses. We've identified it in flowers, by the way, for those of you who are growing only to flower. We have identified it there. And we also know that it can be seedborne. It's the beginning of a new research trajectory, but we know that it can be seedborne. Now we're testing commercial seed lots. We have not found it in commercial seed lots, but coming from harvested grain in our plots, we are finding it.
Hmm.
So we're kind of at a crossroads there. It'll be a few years before we have that data.
Interesting. Okay, so identification. So what are some of the telltale signs of Fusarium in hemp, and are there early detection tools available?
Right now, there are no tools available, but a really nice hand lens or a cheap hand lens or a magnifying glass, if that's what you've got. And so to look inside that head, and I'm just going to use the word head in general for flowers or grain, looking inside, kind of looking at that stem, looking at that rachis in there, and that's where it's going to start. So the more dense the seed head, the more humid it is in there. So if you kind of separate out those flowers or those, it usually starts during flowering. So if you kind of separate out those flowers, just kind of pull it apart, and you can kind of see that fuzzy white growth in there. And that's how we look for it. Now, to identify which species it is, those kinds of things, it's always got to come back to the lab. It's always got to go under a microscope. But you can see that those fuzzy seed head, those fuzzy growth, that mycelia in there. But you're going to have to pull those flowers apart and look inside between those racines.
Okay. And then what do you do after that? You've identified it. Oh, this plant has Fusarium. What's sort of the next steps?
We're research, so we're trying to kill our plants. So our next steps are very different. If it's a
Sure.
grower, I mean, unfortunately, we're at a place where there's nothing that we can do.
Mm.
Right? We don't have a recommendation. We don't have resistant cultivars. Now we're looking at cultivars, right? And we're kind of realizing that some are more susceptible than others. But really, if I'm working with a grower, we're talking about next year. What do we do next year? We're making sure that we don't have a cereal crop in the vicinity, and we're definitely saying not to rotate with a susceptible crop, and not to use a cereal cover crop. And that's something very new, because we thought, okay, we're burning it down and we're crimping it down long before flowering, and that's not enough.
Because that infection is present, even if it hasn't sort of emerged in the flowers.
It's there. Yes, it's there.
Yeah.
Now we have a lot more research to do in that vein. I want to mention before I forget, we did a 2-year study to look at latent infection. So a latent infection is that dormant infection, that time between infection and symptom development. And we were looking at floral hemp. So we weren't going to grain stage. And so we planted hemp every 2 weeks from April until I think September. And then we sampled every week. So in theory, we had We had every growth stage at every week through the season. And we looked at weather data, we looked at growth stage. And so what we found is that we are detecting latent infection 6 weeks or more before symptom development. We're detecting it long before flower initiation. So we're finding it very early in the season, I think as early as May, for those early planted hemp. So remember, that was the same time that wheat was in the field, not in our plots, but in fields. And to overlay weather data, what we're finding out is hemp does become more susceptible as it gets older, as it reaches maturity and as it starts to produce it enters those reproductive phases, but also to layer those rain events. That's when we see huge spikes of fungal DNA within those plants. So we were looking at the DNA in latent infections. So the moral of that story is it doesn't matter when it is. There are times where it's more susceptible. We have more infection and pathogen DNA in that plant, which ultimately becomes diseased. But anytime, infection can happen anytime. Mm-hmm. And that's really scary because in a lot of your cereals that, they're always targeting that anthesis when those anthers start protruding. But with hemp, it looks like latent infection is a lot more common than we expected. And so that's going to take a lot of rethinking on what a recommendation would look like.
Okay. And so when those symptoms, at what stage in the plant's development do those symptoms tend to emerge? Is it during sort of the flowering reproductive stage?
Yes. So symptoms. So disease is what we call symptoms, right? So so the infection is when we can detect the DNA inside.
And is that with qPCR?
Yes, with qPCR.
Excellent.
And anybody listening, that's quantitative PCR. It can detect really low levels of DNA from plant tissue, whatever tissue we choose. And in that case, it was leaves, growing tips. Male flowers, by the way, can become infected, and female flowers. But what was your question? I'm so sorry. I forgot your question.
No, I think we were talking about the latent, identifying a latent infection. So I can just skip to the next question. So,
okay, I'm sorry.
No, that's quite all right.
I went off on a tangent.
I mean, I think just for the purpose. Now I remember what we were talking about. When do the symptoms emerge? At what stage of development of the plant? And I think we said it was during sort of the reproductive stage.
So the later you go into the reproductive stages, of course, induced by a rain event, but the heavier and more dense that seed head or flower head is, the more moisture it's holding. I think there's a lot more symptoms in grain. So there must be something physiological in there. And of course, I don't have any data to back that up. It's just observational. But definitely the bigger and denser and heavier it is at a rain event, or like, when it starts cooling off late in the summer and you kind of get those foggy, dewy mornings, that's kind of the same thing, right? So add moisture and it really gets worse. So the longer you go into the season in general.
Yeah, I think any of us in the Northeast who have tried to grow hemp or cannabis experience that once the fall hits, when you have those dense flowers and those cool mornings, cool wet mornings. That's when the fuzz starts to come out. So going back to the latent infection, can a plant with a latent infection infect another plant, or are they not infectious?
Technically that answer is no. So infection occurs when a spore, Canidia, which is an asexual spore. So when a spore moves, so disease has to develop, the fungus has to sporulate and release that spore, and that spore land on another plant. And when that spore lands on the other plant, they must have moderate temperatures and free water in order for that infection to occur. So a latent infection plant, no. But what we're finding is that if there is latent infection, it means there was a spore deposition event at some point. So we usually have widespread. So maybe one latent infection plant ends up having disease symptoms in 2 weeks, and another one might develop symptoms in 4 weeks. So we don't know. We don't know what those connections are. We have not tracked individual spores. We have not tracked movement.
Okay. Yeah, and you touched on before genetic resistance and that there are no resistant genetics out there right now, but are there certain strains or certain physical characteristics of the plant that can make them less susceptible to an infection?
So we're accumulating data. So I am part of a multi-state group that plants the same group of cultivars across many states. And so we have my working group has tapped into that whole network. So we're going to start trying to understand which of those cultivars, these are hemp cultivars, by the way, which of those cultivars are more susceptible. But what we have found anecdotally over and over is that the more heavy and dense a seed head is, the more disease you'll have. And you've always got those spindly ones with that spacing in between the racemes. There must be more air circulation going there and not having that humidity. That's our guess. So bigger, chunkier seed heads are definitely going to have a lot more disease than the more spindly ones.
And I would imagine it'd be the same for flour as well.
Exactly. Yeah.
Yeah.
So, and I mean, that again, that kind of goes back to those principles of plant pathology, that free water,
Yep.
that indirect, sun not being able to penetrate. And of course, with a flower, flowers are wet and sticky. Whereas when you get to the later season with seed, I mean, they're dry and crispy. So that infection event probably taking place when you've got that higher moisture.
Interesting. Okay. All right. So I guess looking forward here, what are your sort of your next steps or projects that you're working on to better understand this?
So, one of my graduate students, it has transformed the fungus with green fluorescent protein. So he is going to track infection. We're looking at seeds. So we're looking to see whether an infected seed can actually carry the pathogen and if it can move its way up. So those are a couple of the things that we're doing. And we're looking for connections and better identification of DON, the mycotoxin. And it's especially hard in flowers because you've got all those resins that affect the protocols. We're looking at other mycotoxins as well. So we've completed some surveys, those kinds of things. We would like to look more into the animal feed market because it seems like that's where you're going to have that bulk of hemp product that could really be taking some of this Fusarium far and wide and have probably the most effect. So those are kind of our big projects in the future. We're continuing a lot of our field studies, the spore trapping, those types of things. We have looked at, we've done some genetic analyses on the Fusarium that's infecting hemp, and it is the same one that's affecting wheat. So we know that those populations are the same. So that's our immediate look forward. There are a lot more questions, right? What happens if you're using spent hemp, like after the oil's been pressed from flowers? So if you're using byproducts to make animal feed, do you have that movement of those mycotoxins from there? What does stored hemp look like? We've done some stored hemp studies. What does stored hemp look like? And what is that timing, right? So once you dry down those seeds or those flowers to a certain degree, right, less than 15 or 10% moisture, do you stop that DON toxin production? Because that's what's happening in wheat. So can we dry it down more quickly and stop that toxin production? So lots of questions, a million questions, and we're trying to prioritize. I'm an extension professor, so my questions connect directly to growers. So always trying to solve those grower problems first, and then doing the fun curiosity science in between.
Excellent. All right. Well, winding down here, Nicole, please share any other links or studies or resources that you think would be helpful to the listeners. I'm happy to put the links right in the show description so people can easily find them.
Okay, definitely will. We have some new publications in the pipeline right now, so check back on some of these resources and these links that I'm going to send, and that way you'll be able to see kind of as we move forward.
All right, Nicole. All right. Thanks again for taking the time to talk to us about Fusarium and hemp diseases. And
Absolutely.
I look forward to doing this again.
Sounds fun. Thanks a lot. Good to see
All
you.
right.
Bye.